Welcome to Lecture 10.1, in which we will begin learning about genetics. This person is Gregor Mendel, commonly referred to as the father of genetics. We will see how his experiments with pea plants have contributed to our modern understanding of genetics. Our learning objectives are to understand the main Mendelian rules of inheritance, evaluate the results of monohybrid and dihybrid crosses, explain the relationship between genotypes and phenotypes and what dominant and recessive genes are, and use a Punnett Square to calculate the expected proportions of genotypes and phenotypes. What is genetics? Genetics is a study of heredity, which is how characteristics such as our hair color or eye color are passed down from one generation to the next. This image shows a family of 3 generations and how much the characteristics of the parents influences the characteristics of the children. Genes are the sequences encoded in our DNA that code for proteins, and these proteins are ultimately what determines our characteristics. A trait is a variation in the physical appearance of a heritable characteristic, such as having blonde hair or brown hair. As mentioned, Mendel performed many experiments that formed the foundation of genetics. He did experiments with the garden pea plant, which he used as a model organism. A model organism is an organism that is used in experiments to further our understanding of biology. In this case, there are many reasons why the pea plant is a great organism for studying genetics. They have both male and female reproductive organs, and so they are able to self-fertilize. There are also highly inbred varieties that are true breeding, which means that if these varieties self-fertilize, the resulting offspring will always look like the parent. 2 pea plants can also be bred together instead of doing self-fertilization. They also grow quickly and in large quantities, allowing for Mendel to obtain a large number of observations in a relatively short period of time. Another reason why pea plants are great for genetics is that they have characteristics that each have 2 distinct traits. Mendel looked at 7 characteristics in particular, and the 2 traits he observed for each characteristic are shown in this diagram here. We will look at his crosses with flower color first. Mendel began with crossing true breeding violet flower peas with true breeding white flower peas. This first generation is called the parental, or pea generation. What he observed was that all of the offspring had violet flowers, not white flowers. Somehow, all the offspring resembled one parent, but not the other. This first generation of offspring is called the first filial generation, or F1 generation. Next, Mendel let the F1 pea plants self-fertilize. This time, many of the F2 generation still had violet flowers, but some had white flowers instead. The trait that had originally disappeared in the F1 generation reappeared in a subset of the F2 generation. When he quantified the result, about 75% had violet flowers and 25% had white flowers, so there was a 3-to-1 ratio of violet flower pea plants to white flower pea plants. Mendel decided to call the traits that were inherited by all of the F1 pea plants dominant traits, since they seemed to completely overtake the other trait. He called the traits that initially disappeared in the F1 generation, but reappeared in some F2 generation plants, the recessive trait. From these observations, he also proposed that the pea plants possess 2 copies of a trait for each characteristic, and parents pass one of their 2 copies to their offspring. We now know that this is true, and that each characteristic is encoded by genes. The way genes are passed on is through chromosomes. Recall from the meiosis lecture that we all have 2 copies of each chromosome, with one from our biological father and the other from our biological mother. The variation in traits for each characteristic are alleles, represented by the letters next to the chromosomes here. Remember that during prophase 1 in meiosis, crossing over occurs so that some chromosomes become a mix of the paternal and maternal genes, creating a slightly different combination of gene alleles. Recall also that the homologous chromosomes separate into haploid gametes that have only 1 copy of each chromosome, and one of these can eventually be passed on to another generation. Now let's talk about phenotype and genotype. The phenotype is the observable trait of an organism. For example, we can look at these seeds and say that they are yellow or green. The genotype is the gene that contributes to a particular trait in an organism. In other words, the genotype is related to the sequence found in our DNA inherited from our parents, and the phenotype is how that sequence manifests in an organism. In this example, we see that a yellow color is dominant to green because crossing a plant with yellow seeds to one with green seeds produces offspring that all have yellow seeds. We then see in the next generation that about 75% have yellow seeds and 25% have green seeds. However, what's happening at the gene level is a little different. We can represent the gene for seed color with a letter, in this case using Y for yellow because that's the dominant trait. We will assign uppercase Y for the dominant allele and lowercase y for the recessive allele. Since the parental generation was true breeding, that means that they are homozygous, which means that they have the same alleles for a gene. The yellow seed plant has 2 dominant alleles, and the green seed plant has 2 recessive alleles. Since they will both pass on one copy of their genes, each offspring will get a dominant allele from the true breeding dominant parent and a recessive allele from the true breeding recessive parent. Having 2 different alleles for a gene makes the offspring heterozygous. They can also be considered carriers for the recessive genes. We can see that even though they have one dominant allele and one recessive allele, and therefore a different genotype from the dominant parent, the phenotype is the same as the dominant parent's phenotype. When we look at the genotypes of the next generation, we see that unlike the phenotype, which is 75% dominant phenotype and 25% recessive phenotype, the genotype is 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. The homozygous dominant and heterozygous plants all have the dominant phenotype, and only the 25% that are homozygous recessive have the recessive phenotype. A Punnett square allows us to determine how the alleles from each parent can potentially be combined and what frequency each combination will occur at. The way a Punnett square is set up is with the genotype of one parent spread across a row and the genotype of the other spread across a column. For example, let's look at the first cross for pea plant flower color. The violet flower is true breeding and dominant, so it gets 2 uppercase letters. One letter goes in the first row, the other goes in the second row. The white flower is true breeding and recessive, so it gets 2 lowercase letters, with one in the first column and the other in the second column. Now we can combine the alleles from each parent together in the boxes. Since all the rows give an uppercase A and all the columns give a lowercase a, the result is offspring that all have the big A little a genotype and the dominant phenotype. Now let's look at the second cross from the self-fertilization of the F1 generation. The parents are all heterozygous and have one uppercase A and one lowercase a. For the first parent, we will put the uppercase A in the first row and the lowercase a in the second row. For the second parent, which is just itself, we will put the uppercase A in the first column and the lowercase a in the second. Now let's look at the combinations. In the first box, we have an uppercase A from parent 1 and another uppercase A from parent 2, resulting in a homozygous dominant genotype and also a dominant phenotype. In the second box, we have an uppercase A from one parent and a lowercase a from the other, resulting in a heterozygous genotype and dominant phenotype. The same is true for the third box. In the last box, there is a lowercase a from one parent and a lowercase a from the other. This results in a homozygous recessive genotype and a recessive phenotype. In the end, a quarter of the boxes, or 25%, have a homozygous dominant genotype, half have a heterozygous genotype, and another quarter have a homozygous recessive genotype. Looking at the phenotypes, we see that 3/4, or 75%, have the dominant phenotype, and the remaining quarter has a recessive phenotype. This matches the numbers that we saw in the previous slide. Since homozygous dominant genotypes and heterozygous genotypes both manifest as a dominant phenotype, how can we figure out if an organism with a dominant phenotype is homozygous or heterozygous? We can perform something called a test cross. This involves crossing an individual with an unknown genotype with an individual that has a known genotype, which is generally an individual with a recessive phenotype, since it means they must have a homozygous recessive genotype. If the parent with the dominant phenotype was homozygous dominant, all the offspring would have the dominant phenotype. If the parent was heterozygous, and this question mark was a lowercase y, half of the offspring would be dominant because of this uppercase y allele being passed on, and the other half would be recessive because of the lowercase y being passed on. So far, we've been talking about monohybrid crosses, named for creating hybrids in which only one characteristic is different, such as flower color. Mendel also performed dihybrid crosses, in which he crossed 2 plants with different traits for 2 characteristics. In this example, yellow and round seeds are crossed to green and wrinkled seeds. Yellow and round are both dominant and designated respectively by an uppercase Y and R, with green and wrinkled respectively designated by lowercase y and r. This creates F1 offspring that have a heterozygous genotype for each characteristic, seed color, and seed shape. We can use a slightly more complicated Punnett square to see what would happen if the F1 generation was self-fertilized. Each parent must pass on 1 allele for seed color and 1 allele for seed shape. Since it's random which allele for each gene is passed on, there are multiple possible gene combinations. The uppercase Y can be passed with the uppercase R or the lowercase r, and the same is true for the lowercase y. This table shows the result of combining each of the different allele combinations of one parent with the allele combinations of the other. For a heterozygous dihybrid cross, the result is 9 out of 16 having a dominant phenotype for both seed color and seed shape, which is yellow and round. 3 out of 16 had the dominant phenotype for seed shape, round, but the recessive phenotype for seed color, green. Another 3 out of 16 have the opposite, with a dominant seed color phenotype of yellow and a recessive seed shape of wrinkled. The final 1 out of 16 have a recessive phenotype for both seed color and seed shape. An important point is that due to independent assortment, the seed color has no influence on the seed shape, or vice versa. They would be on 2 different chromosomes, so inheriting one does not affect how the other is inherited. In this video, we learned Mendel's experiments with pea plants helped establish the idea of dominant and recessive traits, and that each parent passes on a trait to their child in the form of alleles. The genotype is the allele combination that was inherited from the parents for a gene, while the phenotype is how the gene manifests as an observable trait. We can use Punnett squares to determine the potential combination of alleles from each parent, and how frequent each allele combination is. In the next video, we will learn about exceptions to Mendelian genetics. Thanks for watching.